Back in 2004, my colleague just called me and asked me to take a look at a very serious disease in a vineyard. Then we decided just to have a trip and then to visit this vineyard. We found this vineyard has serious disease. That time we speculate that it's probably caused by viruses. And we test all the known viruses that were discovered in the grape. But we couldn't find a close association of all the known viruses with this disease. It took us several years, to try this new technology, so-called RNA sequencing. That new technology allows us to discover this new virus, which is the first DNA virus that ever reported in grapevine. At the Mountain Grove campus, we have several projects going on in the greenhouses and also in the field, in the vineyard. In the greenhouses, we testing the varieties against these new viruses. In our vineyard, we grow virus-tested 7 grape varieties. I have 2 graduate students, Leanne Herbert is testing the different grape varieties against these viruses. Other students, Shay Honesty, is testing or investigating the genetic diversity of this virus in the vineyards. She visited several commercial vineyards and collected samples and tested the genetic background of the new virus in the commercial vineyard. The new technology, the RNA sequencing, we used to discover this new DNA virus in grapevine can also be applied to discover viruses in any other plants, in fact in humans, in animals. So we use the same new technology, RNA sequencing, to discover those viruses that have not been reported or found in any organisms, including the grapevine. Wild grapes are everywhere in Missouri. After we discovered this new virus in grapevine, we ask whether this virus is existing in the wild populations. The traditional thought is that if the wild grapes contain this virus, maybe it is possible that the virus jumps from the wild grape species to the cultivated grapevines in the vineyard. We tested this new DNA virus in the collections of a particular wild grape species, Vitis rupetrus. And last year, we detect this virus in the Vitis rupetrus, just in one vine. And then we sequence the entire genome of this new virus. Now we are comparing the sequences of this new strain in the wild rupetrus with the original virus we discovered in cultivated grape varieties.